a pleasure to be here. This is a difficult topic. So what is the definition of channelopathy? Well, this is taken from the Merriam-Webster dictionary. You can see this is defined as any various disorder, such as epilepsy, migraine, cystic fibrosis, which is caused by malfunction of ion channel. There are different types of channelopathies. Some are genetics, and these are those we will deal with this morning. And some are acquired, for example, antibodies during some inflammatory disorders, or some medications as well. Now, I don't want you to read all of this, just the title, just to give you an idea of, I'm sorry, can I drink some? So where I put the stars, you have 4 types of channelopathies, which are caused by different ion channels and which cause in turn epilepsies in childhood. What is important here is that you have a number of conditions similar, so different subsyndromes, caused by mutations in different subunits. And we will select some of these for discussion. Now, the clinical presentation is often in the context of epilepsy, with epileptic encephalopathies, which are the most severe forms of epilepsy, especially in children. Then some are manifested as generalized epilepsy syndromes and some as focal epilepsy. But there are also other disorders of neurology manifestations like episodic ataxia, spinocerebellar ataxia, familial hemiplegic migraine, which is a disorder which has a differential diagnosis with epilepsy, and alternating hemiplegia, a form of hemiplegia which can involve different parts of the body. I will focus my presentation of the epilepsies and will mention a little bit the hemiplegic migraine, which has a close resemblance to epilepsy. Now, there are mainly 2 types of channels which are involved. The prototype, the most important for epilepsy, is the sodium channel 1A, which is a voltage-gated sodium channel. There are many other channels with similar characteristics, and ligand-gated channels, like the neuronal acetylcholine receptors subunits. Now, epilepsy. As is possible to see in the slide on the right, you see here the discovery of genes for epilepsy. This is a schematic which represents what has been going on in terms of discovery from 1995, the first gene, and then to 2016 here. You see here there has been an era in the beginning which was defined as the era of channelopathies, where mainly with the support of linkage analysis, so studying large families, so with a selection of conditions which had a limited reproductive disadvantage, what was discovered were situations which were clinically mild. While in the last years, with the support of next generation sequencing, more severe conditions have been discovered. And this is why we have now many genes for epileptic encephalopathies in sporadic cases which have been added to the list. Well, the ion channels are in the first part, but also they continue to be discovered, and many years will need to pass before we have an entire representation of the phenotypic spectrum. Now, this is a review paper we just published with our team, and what we did, among other things, was to review the 13 most important next generation studies in epilepsy, studies using large panels in large series of patients. You can see here that ion channels tend to cluster in the beginning of the schematic, where most patients are represented. And this is a personal series. These are 349 consecutive patients with all epilepsy types, which might have been genetic in origin, so there was no obvious etiology. And what you see here, that 8 out of 10 of the most frequently mutated genes were representing ion channels. So a few genes representing most patients. And then other genes just causing epilepsy in a limited number of patients. And you see here, again, there are some ion channels scattered throughout. So the sodium channels, as I mentioned, are the most important. Voltage-sensitive sodium channels are heteromeric complexes. They have a large central pore-forming alpha subunit and two smaller auxiliary beta subunits. Now, sodium channel 1A, if you look at the inheritance of Mendelian disorders in man, there are 4 conditions which have been associated with it. Dravet syndrome, we will see some of them in detail. Dravet syndrome, which is a form of severe epilepsy starting in infancy. Epilepsy with generalized seizures, which is a much milder form, usually running in families. And febrile seizures, which is a minor manifestation limited to early infancy. And then a few families with hemiplegic migraine. And this is another type of manifestation. Now, this is the most interesting story. Berkovic and Schaeffer in 1997 described large families of individuals where some had generalized epilepsies, some had febrile seizures, some had febrile seizures which were prolonged beyond the usual age for febrile seizures, which is 6 years, 5 years. So they called these febrile seizures plus, because these individuals maintained the propension to manifest seizures with fever even in early adulthood, some of them. Then this other team, 3 years afterwards, identified this sodium channel 1A as a causative in similar families. Then, again, 1 year afterwards, this team in Antwerp, in Belgium, they picked up some very severe cases of individuals having seizures every time they had fever. So they had many, many seizures throughout the time they had fever and beyond, and found that even if they were sporadic, they had the same mutations. So the Dravet syndrome, from the name of the person described this disorder first, was delineated. This is 1% roughly of the epilepsy population. These children have normal development in the beginning, but then they start to have febrile seizures, or, for example, it is usual they have a first seizure after a first vaccination, because vaccination can cause some reaction in the body, some increased temperature. And some of them were classified as post-vaccinal encephalopathies, which is wrong. And they then develop other seizure types. The seizures tend to be severe, and progressively cognitive impairment appears. All seizures are resistant, and other characteristics emerge, especially a high mortality, sudden death, which is still not understood. This is an example of a child at first fever starts to have all these jerks, which you can see. This is not yet a seizure. These are called myoclonic jerks, but they are forecasting major seizures which will eventually appear. And you can see this abnormal jerking. Again. And then a full-blown seizure, which is a major neurological manifestation with convulsive clonic movements. Now, one important thing is that we have the same gene running in families, sometimes the milder phenotypes, and then we have mainly sporadic cases, but not just sporadic, sometimes inherited with very severe phenotypes. So it is important, if you detect a mutation early, can you tell the family this is going to be a severe disorder or not? This genotype-phenotype correlation was attempted initially here, and you see there were some indications. For example, truncating mutations always produce the severe form of Dravet syndrome. The same did missense mutations in the pore-forming region, while, for example, missense mutations in other domains were associated with the milder phenotypes. However, these were rough correlations which did not hold over time. There were, for example, different generations of patients having in the same family much more severe phenotypes, for example, in this proband with respect to the others, even if you had a deletion, which is a very severe genetic defect. And also, it appeared that there were individuals which were born from gonadal mosaicism with unaffected parents, or sometimes very mildly affected parents with mosaicism, low-rate mosaicism, which attenuated the phenotype. But we wanted to understand better this because what is you see here, this graphic on the left, this is the follow-up representation of the developmental quotient of patients, and you see they are normal at age 1, 2 years, and then as seizures appear and they face a severe disorder, the IQ scores drop very much and they stabilize at a level of severe disability from age 6, 7 on. So can we tell the parents after a mutation is detected early, this is going to be very severe, or this is going to be something which is time limited and the child will not have delay? Because mutations don't tell us this. Well, we did a study on 200 consecutive patients with mutations which were detected early. And what is interesting is that the main predictor of outcome was age at onset of seizures. All those whose seizures started within the first 6 months of life had a very severe outcome, and those whose seizures started after 12 months never developed Dravet syndrome. And this is an example. This is the whole series. Overall, 50% roughly developed Dravet syndrome. The others had much milder phenotypes, and this is when onset was after the 12 months. None of the patients developed Dravet syndrome. So here, early clinical indicators are stronger than the genetic indicators. Why we have loss of function mutations which cause hyperexcitability in the brain? This is the main paper which has reconciled these apparently contradictory observations. This is from the Catterall's team. It's 2006. What is important is that they found in the animal model that the SCN1A is mainly expressed in the GABAergic interneurons. So loss of function of GABAergic inhibitory interneurons makes the excitatory neurons hyperexcited and able to generate different types of seizures. But this is certainly easy to understand and makes sense, but studies with iPS derived cells have not produced conversion results, so it's still not well understood what is the basic mechanism. Now, another related condition is the SCN2A epilepsy. As you see here, the structure of the protein is the same. You have 4 domains and the number of transmembrane units delimiting the pores. Now, the story of SCN2A started with these presentations of benign familial, neonatal, and infantile convulsions. These were normal babies having seizures at around 2, 3, 4 weeks and up until 5-6 months. And there were families, there was no reproductive disadvantage. None of them developed seizures afterwards. And this was a clear explanation for the benign convulsions of the first days of life, which are well known to the pediatricians. But the story became more complicated afterwards. The same team pointed out that some of the patients had seizures later in life, a few seizures, were not severe conditions. But then, with the era of NGS, SCN2A was related to severe encephalopathy, so a phenotype which is the opposite to the one we have seen. And the consortium EP4K, which sequenced 4,000 exomes of patients with epilepsy associated with this West syndrome, infantile spasms. Some teams in Japan related the same gene to Ohtahara syndrome, which is the most severe form of encephalopathy with epilepsy in infants. And then other teams related to migrating focal seizures, which is another form of very severe early onset epilepsy. And then, finally, this is almost definitive work by many teams in Europe, as you can see here. They collected a very, very large number of patients with long follow-up, and they made very interesting conclusions. First of all, they found that there were missense mutations with loss of function effect. Some were modeled experimentally, associated with later onset epileptic encephalopathies, and poor response to anti-epileptic drugs, which are categorized as sodium channel blockers. Then they observed that there were truncating mutations, which were only seen in patients with late onset epilepsies, who did not respond to sodium channel blockers. And then, in the end, they also found a subgroup of patients with missense mutations with gain of function effect associated with earlier onset infantile epilepsy, but good response to sodium channel blockers. So 3 different subcategories where genotype-phenotype correlations are relatively good. But there were other descriptions of very severe encephalopathies associated with CN2A mutations, sporadic reports, but they cumulatively produce some interesting evidence, even with severe brain atrophy or with movement disorders like chorioathetosis, like something Dr. Olden will be presenting afterwards, and also forms associated with ataxia. So the phenotypic spectrum is widening, as we have seen before. And then, separately, there were the psychiatrists also doing their studies, and this was a large cohort study of whole exome sequencing in autism, and they found a few patients with CN2A mutations and no epilepsy at all. So it is a gene for autism. And then, of course, as expected, it is also a gene which causes forms of severe intellectual disability, as a different team pointed out. And these psychiatrists went beyond these observations and tried to correlate the different mutations with different neurological or psychiatric phenotypes. And they say that gain of function variants are, in their opinion, observed in epilepsy, while the phenotype of autism is caused by loss of function, severe loss of function. This is an interesting hypothesis, which has still limited experimental evidence. I think more observations will be necessary in the near future. And then, in the end, the last gene of the triplet of genes which cluster in the same genomic area and which cause epilepsy, the third ion channel, which is 8A. As you see, the structure is again the same as we have seen before for 1A and 2A. This is a different kind of encephalopathy. Until now, this is a preponderance of patients who are severely affected, The clinical manifestations are different from those we have seen in the previous two groups. There is no susceptibility to fever. Age at onset of the epilepsy is broader. Seizure semiology is different. We have, for example, infantile spasms here, which are not seen with 1A, and some patients have movement disorders as well. There are not many described, so the entire spectrum is far from being understood. And also, some benign form of paroxysmal movement disorders running in families have been described with a specific mutation which is in the intracellular loop. One important point, which is a large area of research, is that the number of children with SCN8A who die early in life is too high. So there seems to be something not working well in the heart, not just in the brain. This is called sudden death in epilepsy. It's a major medical problem. Is much studied but poorly understood. What is important is that these teams modeled in the animal model two specific mutations who were present in some of the children who died, and found out that they caused arrhythmia in the animal model. We still don't know what's going on in the children, but this frightening observation, for example, which we made, is really ringing something more studied are advised in these children. Look at this, what is going on here. This is a child who has electrocardiographic monitoring here, and also has an electroencephalographic monitoring. This is electrical activity in the brain going on. Is not good, but it's not bad either. At some point, a seizure appears here. Is a seizure with stiffening of the body, and the heart stops beating. And here there is about 15-second absence of heartbeat, which is really a frightening manifestation. So there are studies going on in different parts of the world trying to understand why these children are so fragile. Another category of channels which are important for epilepsy are potassium channels. Potassium channels, as you can see here, have been associated with different forms of epilepsies, and also have been associated with other manifestations like episodic ataxia, which is a form of paroxysmal ataxia, developmental delay, and diabetes as well. Now I will discuss a little bit the sodium channel Q2, which has been associated with two opposite phenotypes. So the first phenotype here was described in 1998, and again were benign seizures starting early in life and running in families. So like we have seen for CN2A, they have similar manifestations early in life, short seizures for a few weeks and then complete remission. After these observations, there were others, however, underlining that some patients in the same families had a severe encephalopathy, and this is a child whose mother had benign seizures, and this is the child. These families were easy to collect. Some of these mutations were modeled, and some teams pointed out that what was happening clinically was the consequence of upper insufficiency with loss of function caused by the specific mutations which caused these phenotypes. But we move to the era of next generation sequencing, and again, same reasoning I have done before. Sporadic severe cases were studied, and it was found that different mutations of the same gene were associated with very severe encephalopathies. This is an EEG with suppression bursts, which is the worst EEG you can have in a child, with phases of suppression of electrical activity in the brain. And also some had atrophic changes in the brain. These were onset of intractable seizures in the first weeks of life. But what was bizarre is that in spite of this stormy, very severe onset, most of the children became seizure-free after 3 years, but they were left with severe developmental delay. So mutations causing these encephalopathies were modeled again, and what this team demonstrated in a very elegant fashion was that most of these mutations cause a drastic dominant negative effect on the wild type allele, and that was the mechanism. But other teams have demonstrated the opposite. For example, this is a very elegant study by Talia Tela in Naples, and they demonstrated that the early onset epileptic encephalopathy may also be caused by gain of function mutations. So here again, it is not easy from the beginning to tell what is going to be the phenotype. An interesting observation, however, is that these patients tend to respond to sodium channel blockers, probably because of the interaction between sodium and potassium channels on the cell surface. And if they are treated promptly and seizures go into remission, the chances of having a good neurological development are much enhanced. Then the last category I wanted to approach of genes for epilepsy, ion channelopathies, is a fascinating one. These are the subclass of nicotine acetylcholine receptors, which have been associated, all of them, with the same type of epilepsy, even if the semiology of manifestations may be very variable. This is the first very large family, as you can see from this pedigree, which was collected in Australia and was the very first gene associated with isolated epilepsy by the St. team. This is a subunit receptor alpha 4. Since then, another two subunits have been associated with a similar phenotype, and we know this is a type of epilepsy which is running in families, is limited to sleep. These patients have seizures only when they are asleep. Penetrance is 75%. There may be marked intrafamilial variation in severity, and this is often overlooked because these manifestations, if they are mild, as we will see in the video recordings, they are misdiagnosed as behavioral episodes or sleep disturbances, not epilepsy. Before we see the videos, we have to understand that this abnormal activity of the brain seems to originate from very specific areas, in particular the frontal, the cingulate gyrus, where you have a number of manifestations, but which is strongly related to the emotional part of the manifestations, and the frontal orbital gyri, where some automatic motor manifestations are encoded. These seizures were called in different ways. They were called hyperkinetic or complex motor or hypermotor, and this latter term is the one which tends to be used lately. And this is an example. This child, young man, is asleep, wakes up, and he starts to do these bizarre movements, which have a pattern. It's not just chaotic. He's entirely conscious, tends to react, and then the episode is finished and goes back. So this is not a major episode, but if imagine having this 10, 12, 15 times per night. Manifestations you have seen are hypermotor, because there is an excess of motion, but there are other manifestations as well. For example, this is the second subunit, is alpha 2, which was related to the same type of epilepsy related to sleep, but here the manifestations were mainly emotional, and this is an example. So waking up suddenly with a lot of fear, even if maintaining consciousness. And some are much milder. This is called paroxysmal arousals. So these patients wake up, they just sit in the bed, look around, or may even walk and then get back to the bed. This, for example, is what was found in the only family described until now with loss of function. And this is an example. It might not be working. Okay. Child is asleep, he just sits on the bed. Changes position, is a bit tremulous. And then goes back to sleep. You will never tell this is an epileptic seizure, but EEG shows it. And this is his brother. He's sleeping with his mother. Wakes up. He's a little bit confused. Has some automatic movements. It doesn't respond to questions. So now a few words, which will be the introduction for, I think, the next speaker. As I mentioned before, there are some ion channels associated with other manifestations, in particular sodium channel 1A and potassium channel alpha 1, which have been associated with episodic ataxia, which are episodes of sudden cerebellar signs. The patient becomes unstable, tremulous, is unable to maintain a normal posture, and this is a very disabling and strange clinical manifestation. And then the hemiplegic migraine. Hemiplegic migraine, as you can see here, is there are three subtypes which have been described and have been associated with different ion channels: calcium channel 1A, the alpha subunit of the sodium-potassium pump, the ATP1A2 gene, and with the gene we already mentioned, sodium channel 1A. This is the less important of the three. Most of the patients have mutation on either of these two genes. The attacks are like attacks of severe migraine, but the aura, which means the visual manifestations or the motor weakness, is much pronounced and may reach very severe levels of disability, which can last for hours. It is a very challenging disorder, which has little benefit from treatment approach, and this is especially at its onset a cause of hospitalization with emergency unit, and these patients are thought to have encephalitis or a stroke, are studied for actually no reason, because this is a reversible manifestation. Now, as you see, these are the three subtypes. The calcium channel 1A, there are usually missense mutations and are gain of function, and they lead to increased calcium influx, which has the effects of enhancing the glutamatergic transmission. This is supposed to be the cause of the manifestations I mentioned. Then we have mutations in the ATP1A2 gene, which produce various variable extents of loss of function of the alpha 2 sodium potassium ATPase, and some studies have been carried on. These patients have usually nothing interictally, but ictally some alterations have been inconsistently found in angiographic studies, for example, in MRI studies. But this is something very difficult to perform. What is easy to perform, which can help recognizing an attack of hemiplegic migraine, is an EEG during the attack or just after the attack. It is not an EEG indicating epileptic activity, but is indicating that one area of the brain is producing very slow activity, which is a non-specific sign of suffering, but which becomes relatively specific in this condition with respect to the others which have differential diagnosis. So to conclude, certainly I could not deal with all the ion channels, which you have seen in the several slides, because there are hundreds of them. But we can say that in addition to the fact that the neurological channelopathies cause many different neurological diseases, these tend to be usually autosomal dominant, even if in the most severe expressions they are only seen in sporadic patients. The main manifestations are paroxysmal, as you have seen, but there may be impairment of function which become fixed over time. Function in patients with epilepsy is cognitive function. And some of these encephalopathies, as you have seen, when they are manifested with frequent seizures, they always carry cognitive impairment of different kinds. It is now our interest opening to their role in a psychiatric area, especially autism. These situations are individually rare, but collectively they are relatively frequent, as you have seen from the graphic of the reports of the NHS studies. The implications for genetic counseling and treatment are very complicated. And counseling a patient, for example, with a CN1A mutation and febrile seizures about a child is very, very difficult. You can never tell whether the same mutation will produce the same mild defect or a much more severe phenotype. And treatment implications, there was no time to discuss, but I can summarize in a few words. Treatment is relatively ineffective. It is very difficult to act on patients with a channelopathy based on a proof of concept which has been obtained and in studies on animals, for example, and get some benefit. What has improved over time is that we know what not to do with them. For example, we know we should never use a sodium channel blocker in a patient with SCN1A because we know that they can worsen, and this has been described. But then in another channelopathy with a similar effect, the same medication may work. So we are very far from understanding the real mechanisms, the basis of these disorders. So thank you for your attention.